Hi, I'm Charline Walker from the lab of Charles Kimmel at the University of Oregon, the Institute of Neuroscience.
And I'm Gregory Walsh. I work in the lab of Cecilia Moens in the Division of Basic Sciences at the Fred Hutchinson Cancer Research Center.
Today we will show you a procedure for making gynogenetic diploid zebrafish by the early pressure method. This method uses hydrostatic pressure to depolymerize microtubules in an egg fertilized with UV irradiated sperm. Done with the right timing, this prevents the expulsion of the second polar body, generating a diploid embryo, whose homologous chromosomes are the two products of meiosis I. This technique was worked out in the early 1970s when I worked in the lab of George Streisinger at the University of Oregon.
In our lab, we use this procedure in 2-generation forward genetic screens. The gametes of F1 mutagenized fish can be screened directly for recessive mutations that cause developmental phenotypes in homozygous gynogenetic diploid offspring. Since zebrafish exhibit high genetic interference, we also use this method to map mutations to chromosomes and to estimate how far these mutations lie from the centromere.
So let's get started.
During the late afternoon the day before the experiment, prepare for sperm collection by separating males and females to be squeezed.